Hi, welcome dear students. So today's topic will be biomolecules and we will be dealing with the carbohydrate part. Okay, so straight away, what are carbohydrates? Carbohydrates. The name says carbohydrate. So there would be something involved with the carbon, something hydrate. So water. Okay, that means H2O. So CH2O. Very lucidly, we can say this is the thing, carbohydrate. Now, if we go to more technicality, the proper definition of carbohydrate, they are aldehydes or ketones, ketone derivatives of the polyhydroxy alcohol. If the definition seems a bit tough, we'll again, I'll first repeat, okay? Carbohydrates are aldehyde or ketone derivatives of polyhydroxy alcohol. Polyhydroxy alcohol means there would be more than one hydroxyl groups, okay? And there would be either a ketone group or an aldehyde group, okay? So they can either be aldoses or ketoses, as we will see later on. And as I said, the definition started with carbohydrate. So the ratio of H and O would be 2:1. Okay? Understood? So again, there can be additional some other elements like the nitrogen, like the phosphorus, the sulfur. Okay, those are certain derivatives of the mother molecules. Okay, but overall, a carbohydrate should have, so mandatorily should have carbon, hydrogen, oxygen, carbohydrate. Okay, this is how we just lucidly start. I'm telling you, carbohydrates will be complex, but I won't jump a single step. I would try to lucidly explain everything in a layman's language as much as possible. Okay? So now what are the types of carbohydrates that we get basically? The types of carbohydrates are, as we divide them into monosaccharides, oligosaccharides, and polysaccharides. So why are these? Why are these terms and why those mono, oligo, and the poly? So mono, basically, if you consider a carbohydrate like a pearl necklace, okay, this monosaccharides, they are the individual units, they are the individual pearls. Okay, and then those pearls would be joined by a string. Each part of the string we call a glycosidic bond. Okay, and that bond totally makes up the pearl necklace. You join the pearls one after another, you seal them one after another using the string, and ultimately you get a pearl necklace. Okay, so these monosaccharides are our basic units. Now, these monosaccharides, as I say, they can be either aldoses or they can be ketoses. Means they can either have aldehyde group or they can have ketone groups. Okay. Now, based on the number of carbons that they have, we would see that they can be trioses, heteroses, pentoses, hexoses, and heptoses. Okay. Now, what are these? Trioses are 3 carbon compounds. Heteroses are 4 carbon compounds. Pentoses are 5 carbon compounds. Hexoses, 6 carbon compounds. Heptoses, 7 carbon compounds. Okay, let's quickly look at a few examples. The very basic trials that we talk of. There are 2 types. One suppose we go for an aldose, the glyceraldehyde. Okay, and let's go for a ketose, the dihydroxyacetone. Okay, we will go to each and every example and also with structures. The important examples will go with structures and we'll explain each and everything. So now glyceraldehyde and dihydroxyacetone phosphate. Okay, so these are basic trioses, 3 carbon compounds. So these are what we can essentially say, these are the most simple carbohydrates. Okay, then tetroses. Tetroses, we can have erythrose. Okay, then pentose, we can have ribose. Then hexose, we can have glucose. Heptose, we can have cetoheptulose, which you get as an integral part in plant photosynthesis. Okay, so these are different small pearls, small monomers. Okay, now what about the oligosaccharides? Oligosaccharides are more than 1 units, but not in huge numbers. Technically, as we go for proteins as well in the peptides, so oligo generally would mean the monomer between 2 to 10. Okay, monomer between 2 to 10, we generally term them as oligosaccharide. So here, suppose under oligosaccharide, we get a disaccharide. Disaccharide, we get a trisaccharide, we get a tetrasaccharide, some of the examples I'm telling you. So disaccharide, very, very important example you'll see in the digestion chapter, and that is our sucrose, that is our maltose. Okay. That is our lactose. These are very, very important examples. Okay, then in trisaccharide, you may have raffinose. In tetrasaccharide, you may have stachyose. So some of these common examples. Okay. So what about the polysaccharides? Polysaccharides, we can again divide into homopolysaccharides, heteropolysaccharides, and conjugate polysaccharides. Okay, so what about homopolysaccharides? Homopolysaccharides means, suppose you have a pearl necklace with only white pearls. So all pearls are of same types. That makes your homopolysaccharide. Heteropolysaccharide. Your one pearl is a white pearl. Your one pearl is a pink pearl. Or let's say one pearl is a black pearl. Okay, whatever. And so all the different monomers you can expect in a heteropolysaccharide. Okay, so on polysaccharide, very easy. We can see that is the starch like the gin, etc., the cellulose, etc. So all these are homopolysaccharide. Heteropolysaccharide, let's say agar. Okay. And among the conjugated polysaccharides, we'll see lots of examples there. And we will find glycoproteins there. We will find the mucoproteins there. We'll find glycolipids there. So we will deal with this later on. Okay, so this is a very basic classification of the carbohydrates. Okay, based on their numbers, based on their composition. Fine. So we'll move on to the next one. Next, we are more, or what we are concerned about, what actually confuses students about that is the different isomerism part of the carbohydrates. That we'll go deeper into the structures. Okay, so isomerism in the carbohydrates. So what type of isomerism do we find? We will find, already we have talked about some isomerism like the aldoses and ketoses. Okay, so aldose, ketose, isomerism. Then we find something like DL stereoisomerism. Or we call them enantiomers. Okay. Then we can have epimers. We can have something like anomers. We can have something like pyranfuranmers. All this, I promise you, I'm going to explain in most lucid detail, and that too, not jumping a single step. Okay, so in case you find somewhere I have been a bit fast, you pause the video, you go back, and you see repeated number of times. Okay, and at the end of the video, I will also ask you some questions based on whatever concepts we have discussed, so that you can test yourself how you have understood the thing. Okay, so let's first go to the aldose ketose. So I said that there are two very basic types of aldoses and ketoses, the examples that I gave. One is the glyceraldehyde, one is the dihydroxyacetone. So since they are, I said the most basic thing, basic is the fundamental, the smallest one. So the smallest, what did we see last time? We saw they were trioses, right? They were trioses, means 3 carbon compounds. So 3 carbon compounds will drop first 3 carbons. Okay, will first drop 3 carbons. Now, here, as I said, that there will be this one will have an aldehyde group, and this will have a ketone group. So aldehyde and as the definition says that they are polyhydroxy alcohol, so more than one alcohol. More than one alcohol. Okay. So see here, we have got our glyceraldehyde. Glyceraldehyde. Okay. And here, we have dihydroxy. Again, it's a polyhydroxy. Polyhydroxy only makes our alcohol, our carbohydrate. Okay, so it's a dihydroxy here, dihydroxyacetone. It is the word dihydroxyacetone. And look at this very carefully. I said they are aldose ketose isomers. Let's count the number of carbons. 1, 2, 3. 1, 2, 3. So both have 3 carbons. How many hydrogen? 1, 2, 3, 4, 5, 6. Here, 1, 2, 3, 4, 5, 6. So hydrogen numbers also match. How many oxygen? 1, 2, 3. Here also 1, 2, 3. So what did we see here? So C, M, H2N, O-N. Here N equals to 3 for both. For both, this is the one, right? This holds good for both. Only and only we can have one exception in this formula. Suppose we have a few poles. Actually, it's a hexose. Actually, it's a 6 carbon. Few poles have C6. H. Let's draw the structure of fucus. Find out. No, I don't want to jump any step, I said. So we'll have one very idea. Okay. And also. At the end here, instead of CH2OH, we have CH3. That's CH3. Understood? So actually, one oxygen is missing. So it will be H12, fine. But O5, not O6. Otherwise, for all hexoses, we will find C6H12O6. Okay. So we are going back here. So CnH2 how it's good for both of these. All is good for both of these, no problem. Now, so our aldose ketose isomerism holds good, right? We have understood what is an aldose, what is the ketose. Now let's come to something in the glyceraldehyde. So we'll start with our second concept, that is DL stereoisomerism. Okay, DL stereoisomerism. Now, what is considered as a reference frame is the penultimate carbon of the diglyceraldehyde. Of the glyceraldehyde, let's say glyceraldehyde. Penultimate carbon of the glyceraldehyde. First of all, which is the ultimate carbon? Means the ending carbon. The ending carbon is this one. Okay. So by IUPAC nomenclature, this is number 1. So this is number 2. So this is the number 3 carbon. So this is the ultimate carbon. So who is the penultimate carbon? Penultimate is the last to the is the previous one to the last. So this carbon, this is the penultimate carbon, right? Penultimate carbon. So number 1 concept, we now identify something called the penultimate carbon. Okay. Concept number 2, there is something called chiral carbon or asymmetric carbon. So what do we call a chiral carbon? Chiral carbon is something which holds 4 different things, 4 different side groups, functional groups, whatever you say, 4 different things in its 4 balances, in its 4 hands. Okay. So if we see our glyceraldehyde, which one is the chiral carbon? Let's see right from carbon number 1. Carbon number 1 has 2 arms engaged to oxygen. So all 4 arms are not different. So number 1 cannot be chiral carbon. Let's look at number 3. Here, 2 arms have hydrogen. So number 3 also cannot be chiral carbon. But let's look at number 2. One hand, it is H. One hand, it is OH. One hand is an aldehyde group. One hand is CH2OH. So basically, this carbon number 2 is our chiral carbon. Okay, this is our chiral carbon. The two concepts we can take from here. One is we now identify the penultimate carbon. Number 2, we now identify the chiral carbon. Okay, and the beauty of the chiral carbon is that there can be rotation of the functional groups around these chiral carbons. So you can have either glyceraldehyde with this structure, okay, or glyceraldehyde with keep the structure, keep the orientation of functional groups same in the non-chiral carbons. Okay. So these orientations we are going to keep the same. The rotation can be around the second one. So it can either be this form or it can either be this form. This form we call D form. Okay, the T enantiomer, T stereoisomer. This is our D-glyceraldehyde. Okay, this is our D-glyceraldehyde. And this is our L-glyceraldehyde. For fun, it may seem to be pretty easy, but when we start to compare other monosaccharides, keeping a reference frame of these, then it starts to get a bit difficult. I mean, initially, but after this class, we won't have any problem. Okay, so let's see. Why am I, which thing am I calling D? When in the penultimate carbon, I repeat, when in the penultimate carbon, the OH is on the right, H is on the left, then we call this the D form. If in the penultimate carbon, OH is on the left and H is on the right, then we call it the L form. Just plain and simple, try to keep it. You'll understand this. Okay. Now, whichever carbohydrate we come against, whichever monosaccharides we come against, and if they ask you, tell me whether it is D form, the D stereoisomer, or the L stereoisomer, you will compare that, keeping glyceraldehyde as the reference frame. Okay, keeping glyceraldehyde as the reference frame. So straight away, we go to a complex one. Okay, straight away we go to a complex one. Like, suppose we're talking of glucose. Okay, so let's see. So glucose structure is this. Glucose is a hexose. 1, 2, 3, 4, 5, 6. Glucose is a hexose. Done. First, before going to the answer whether glucose is a D-glyceraldehyde, whether this form is a D-glyceraldehyde, the D-glucose, or whether this form is a L-stereoisomer, we will first try to identify the chiral carbons in glucose. If now I asked you, how many chiral carbons are there in glucose, or identify me that chiral carbon in glucose, that asymmetric carbon in glucose. Okay, so you can just try to pause the video for a minute You try to identify them. I am taking 5 to 10 seconds pause. So I'll give you the answer after this, but you pause the video right at this point and you try to figure it out. Which of the carbons here that I'm mentioning, it's chiral carbon in the glucose. So let's say this is carbon number 1, this is carbon number 2, carbon number 3, carbon number 4, number 5, and number 6. So now you identify for me which one is the chiral carbon in glucose. Which one or which ones? Okay, take your time, pause the video, and try to write the answer in your copy. With reason, with proper logic, you try to answer and see if your answer matches with my answer just after 5 seconds. Okay, so here we are. Here, this one, carbon number 1, 2 hands are engaged to oxygen, so no chance. Carbon number 6, 2 hands are engaged to hydrogen, so no chance. What about carbon number 2? 1 hand H, 1 hand OH, 1 hand aldehyde, 1 hand this entire group. But definitely carbon number 2 is chiral carbon. What about carbon number 3? 1 hand OH, 1 hand H, 1 hand this entire group. 1 hand this entire group. This entire group. So definitely carbon number 3 also our chiral carbon. Carbon number 4, 1 hand H, 1 hand OH, 1 hand this entire group. 1 hand this entire group. So carbon number 4 also chiral carbon. Carbon number 5, 1 hand H, 1 hand OH, 1 hand CH2OH, 1 hand this entire group. So out of 6, C2, C3, C4, C5, these 4 carbons are chiral carbons. Understood? These 4 are chiral carbons. So what can we achieve out of this? What can we achieve out of this? We can achieve. Let's draw the glucose once more. If now somebody asks you which form of stereoisomer is this glucose? So you can identify the molecule that this is glucose, fine. But what is the stereoisomer here? So we go back to our glyceraldehyde, keeping glyceraldehyde as our reference frame. We will go to the penultimate carbon of the glyceraldehyde. So in the penultimate carbon of the glyceraldehyde, if the penultimate carbon has OH on the right, we call it D. If OH is on the left in the penultimate carbon, I'm making a bit strong, bold structure here, so it avoids any confusion. So OH on the right here, so D. OH on the left here, so L. Now, what is the penultimate carbon in glucose here? penultimate carbon in glucose here. So this is our functional group made the aldehyde one, right? So this is C1. So this is C6, our ultimate carbon. Which is the penultimate one? Penultimate one is the C5. This is the penultimate carbon. Right? And in the C5, what is the orientation? Where is OH? OH is on the right. OH is on the right, just like our D-glyceraldehyde. So this is our D form of glucose. Understood? This is our D form of glucose. Oh, what about our L form of glucose? So, sir, you just rotated it like a mirror image. You just rotated the groups around the chiral carbon and you got the L form. So let's rotate this one. Let's rotate this one. Then we will get the L form. No. That's a mistake. Right? If you have to rotate to get the L form, remember, L stereoisomers are, remember this, non-superimposable mirror images around all chiral carbons. Non-superimposable mirror image around all chiral carbons. What are your characters? First of all, non-superimposable. Second, there would be obviously mirror images. And third would be rotation should be around all chiral carbons. Rotation around all chiral carbons, not just one. Not just one. Okay. You know what would happen if there is rotation just around one? It is a totally different type of isomerism. It becomes epimerism, not your DL stereoisomerism. Okay. Of all the isomers that you have here, let's look at the characters of the compounds. Aldose, ketose isomers. What do you see here? This is aldose, this is ketose. Do you see they are same compound or different compounds? Obviously different compounds, right? So aldose, ketose isomers, they are different compounds. DL stereoisomers. What I'm saying, D-glyceraldehyde and L-glyceraldehyde. Am I saying D-glyceraldehyde and L-dihydroxyacetone? No. D-glyceraldehyde, L-glyceraldehyde means both are same compound. So in our DL stereoisomerism, compound remains same. But in aldose ketose isomerism, compounds become different. In our epimerism, compounds become different. Here, if we say that, yes, sir, I can just rotate this one. If you rotate this one, not only you will not get L-glucose, actually you get something which is not glucose. It totally turns into a different compound. Understood? It becomes an epimer of glucose. So how to make actually L-glucose? For L-glucose, you have to rotate all the 4 functional groups to make mirror image of this. So let's make it here. So we are not touching our non-chiral carbons, okay? We are not touching our non-chiral carbons. I'm repeating. Do not make this mistake. Never remain safe. let's now make the rotation around all. So this one becomes, right? So this is, let's number them, then it would be easy. So this is 1, this is 2, this is 3, this is 4, this is 5, this is 6. So here also 1, 2, 3, 4, 5, and now 6. So let's rotate around all of C2, C3, C4, C5. So it becomes, now this C3 becomes HOH. C4 becomes OHH. C5 becomes OHH. Right? So we have rotated around all 4 of these. Rotated around all 4 of these. And we have got our L-glucose. L-glucose. Understood? So for everything is clear. Right? Any doubts so far? Anything? So a very quick recap. First of all, iodose ketose. How many isomers? Iodose ketose isomers, DL stereoisomers, epimers, anomers, pyran furan isomers, of which are same and different compounds. Iodose ketose different. DL stereoisomers, same compounds. Epimers, different compounds. Anomers, same compounds. Pyran furan isomers, same compounds. We will see each and everything, okay? I promised I won't jump a single step. Fine. So we have got our D-glucose, L-glucose. We have got, we now know properly the different D and L forms, right? D and L forms. So now it's up to you. Now I will give you one problem here. So you can test yourself. I am drawing the D structure of galactose. Another hexose I'm drawing, okay? D-galactose. Let's say move 3, 4, 5, 6. Okay, let me make it up for you. Okay, let me make plus. So, first of all, I told you this is galactose. Fine. Now identify what. First of all, you tell me what is this? Is this an aldose sugar or a ketose sugar? Very easy. It's an aldose sugar. It has aldehyde group here. So you got your point. Next is, now you tell me whether this is a D form or an L form. Whatever form it is, you now convert it to its other stereoisomer. First of all, you tell me whether it is a D form or an L form. Pause the video, think yourself, see the structure, and now think yourself whether this is D form or L form. You got your answer. It is D form, right? Why D form? Again, penultimate carbon is our C5 here. On which side is the OH? OH is on the right. Where did we have OH on the right? We had OH on the right for The penultimate carbon, not diglyceraldehyde. I'm telling you, every time we will compare with the diglyceraldehyde. Okay, so in the penultimate, where did we have it? In the D, OH is on the right. So here also, OH is on the right. Fine. So if OH is on the right, this is our D-galactose. Okay, now you make it. Now you make the L form. Again, pause the video. In your copy, write the L form and see if it matches with mine. Okay, if it matches with mine. So I am drawing the structure after about 5 to 10 seconds. And while you pause the video and you draw the structure yourself. So here we are, 6 carbons. So C1, C2, C3, C4, C5, C6. Okay. And what did I say? To achieve the L form on the other stereoisomer, we have to rotate all the functional groups around all chiral carbons, right? So we are rotating this one as which H, this one as H, OH, this one as H, OH, this one as OH, H. We found total rotation, right? Okay, considering these two, we have got non-superimposable mirror images, right? Okay, so this is our L-galactose. I hope after this, you will not make any mistake about the D and L forms. Right? So our DL stereoisomerism done. Next comes, as I said, epimerism. Epimerism, if there is a change around a single asymmetric carbon. Around that single asymmetric carbon, if there is a change, then we call that epimerism. Okay, and it should happen beyond our trials, obviously, because from the trials, one single change means it is only being the DL stereoisomer. Okay, so beyond that, so more than from the tetroses, if we find that there is a change in a rotational position of the functional groups in only one of the asymmetric carbons and not all, then we can have epimers. Okay. To understand the epimers properly, I will go to something. We will see different examples of the monosaccharides, different examples of tetroses, of the pentoses, of the hexoses. Okay? And let's see what happens. I told you I won't jump a single step. Okay? So let's see. We'll build from here. We will build from what? We'll build from, okay, before that, let me just tell you, this structure that you are seeing, since these carbons, drawing them becomes so repetitive, actually we later on draw them like this. Because the non-chiral carbons don't change anything. So non-chiral carbons, we are drawing like this, okay? CHO and CH2OH. And whatever with the carbon numbers, if it's a hexose, then there would be 1, 2, 3, 4. So this point is our C2, this point is our C3, this point is our C4, this point is our C5. Like this we draw. Okay. So our D galactose now becomes actually HOH, HH, HH, HOH. This is how we draw. Okay. This is our D galactose. Understood? This is how we will draw in this one. So where do we start? We start with the diglyceraldehyde. So CHO, CH2OH. Since it's a D form, so in this page we will draw only the D forms. Okay. That would not complicate things. We'll draw only the D forms. So this is what? This is our D-glyceraldehyde. Okay. D-glyceraldehyde. Now we will step up. Step up with what? With one more chiral carbon. Okay. So we are adding one more chiral carbon. That's 1C. Since the chiral carbon, I'm just adding a star. So what can we have? We can have this CHO, H2OH, HO, 2OH. Glyceraldehyde structure remains the same, so HOH. And since penultimate carbon added, all I said would be D, the penultimate carbon would always from now on be like this. Okay. So this is also D form. So, but I said I have added one more chiral carbon. So one carbon being added here. And since it's a chiral carbon, there can be rotation around the groups. So we can have two orientations, two forms, two types of stereoisomers. One can be in this form, HOH. One can be OH, H. Okay. So what is this? This is our B-erythrose. And this is our B-threose. I'm emphasizing on two types of isomerism again, or rather, let's say through all three types. So this one is an aldose, this one is an aldose, this one is an aldose. We are not talking about any ketones in this page. So all are aldoses. Now, what about the enantiomer? See, all are having, in the penultimate carbon, all are having OH on the right. In the penultimate carbon, all are having OH on the right. All are having OH on the right. So all are T forms. Now, even in the D forms, there is a rotation around one asymmetric carbon between D-erythrose and D-threose. And what did you just see? The compounds changed. The compounds changed means what? They are epimers of each other. Isomerism, compounds are different. Okay. And there is rotation only around one asymmetric carbon that we have seen, right? Only around this C2, only around this C2, there is the rotation. Okay. Let's again add one more chiral carbon to each of these. So I'm adding one more chiral carbon plus 1C. And adding one more chiral carbon to both of these plus 1C. Because obviously C would come with their groups HOH. I'm just writing that we have added one more carbon. So now let's keep the structures intact. HO, H2OH, CHO, H2OH, sure, H2OH, sure, H2OH. Okay. Now, we will keep the erythroid structure intact. So erythroid structure to be kept intact. H, OH, H, OH. Similarly here, H, OH, H, OH. Okay. Now, I added one more chiral carbon here, here. So, one form can be HOH, but its epimer can be OHH. And see, we have rotated around just one asymmetric carbon. So they have become epimers. So they have become different compounds. This is our D-ribose, whereas D form, obviously, see the penultimate carbon, OH is on the right. Here also penultimate carbon, OH is on the right. So definitely D form. But this is our D-ribose, and this is our D-arabinose, different compounds. They are epimers of each other. Epimer at what? Epimer at C2. They are epimer at C2. Now, keeping the structure of trios intact, so we will have OHH, we will have HOH, we will have OHH, we will have HOH. We added one more chiral carbon here. So orientation can be HOH and OHH. So they also, the difference is one rotation at one chiral carbon. So they are epimers of each other, but definitely different compounds. This one is our D-xylose, whereas this one is our D-lexose. Okay, different compounds. D-xylose, D-lexose. So they are epimers at what? Epimers at C2. They also are epimers at C2. Okay. Understood? Now, let's add one more asymmetric carbon to each of these. So here also I'm adding one more asymmetric carbon. That's 1C. See, I am not jumping a single step, I promised you. We will make up each and everything. So let's draw the ribose structures. Choose each to each. Everywhere. Let's first draw the structure. Okay, so let's draw the ribose structures. H, OH, H, OH, H, OH. H, OH, H, OH, H, OH. We added one more chiral carbon here. So rotation can be resulting in H, OH and OH. So they've rotated at one chiral carbon only, so they are epimers of each other, and since they are epimers, they are different compounds. What forms? Obviously, as I said, all of these in this page will be D forms. Penultimate carbon remains unchanged, so there would be D. This is our D altrose. D allose. This is our D allose, and this is my D altrose. Okay. Again, let's keep the Arabicos framework intact in both. This will be OH, OH. So we added one chiral carbon here. Rotation results in H, OH, back, OH, H. Again, they are epimers of each other. Our very own D-glucose. Our very own D-glucose. Okay. And it is epimer with what? This one at C2. At C2, this is our D-mannose. Okay. Mannose we don't really find, so mannose you will find in our conjugated, our egg albumin. Okay. So the mannose is very important carbohydrate. Okay, also in cell wall we find mannose. So D-glucose and D-mannose, epimers of each other, different compounds. Okay, let's bring the xylose structure. 1, 2, 3. 1, 2, 3. So H, OH, OH, H, OH. And then again same H, OH, H, OH. So but also one carbon we have added. Here also one carbon we have added, carbon. So after addition, two possibilities. It can be H, OH or OH, H. So again, epimers of each other, they become D-glucose, G-idose. Okay. Again, lixose, obviously they are epimers of each other. D-glucose and D-idose are epimers of each other at C2. And let's bring the lixose finally. 1, 2, 3. 1, 2, 3. So OH, H, OH, H, H, OH. Okay. OH, H, OH, H, H, OH. That's okay. We have added one more chiral carbon here. Okay, so this becomes HOH, but this becomes OHH. Okay. This is our D-galactose. And this is our D-talose. So D-galactose and D-talose again, they are epimers at C2. Epimers at C2. But now, one more question that comes from here. One more question, that is, which of the compounds, they can give you different compounds, 4 options given. Which of these compounds are epimers of glucose? Okay, so let's say the question comes, which of these compounds are epimers of glucose? Option 1 comes mannose. Option 2 comes galactose. Option 3 comes allose. Option 4 comes all of the above. Okay. Epimers of glucose. Let's see. Mannose, we have already discussed. Mannose is an epimer at C2. But what about allose? Allose is also an epimer of glucose at C3. See, only one rotation. Only one rotation between these two. Right? Only one rotation between these two. And this also. So allose is also an epimer of glucose. Okay. What about galactose? Galactose. So mannose is epimer at C2. Allose is an epimer at C3. Galactose. See here. C4. Only one change. Sorry, this galactose. C4, only one change here. Right? Between glucose and galactose, only one change at C4. Otherwise, the entire structure is intact. Galactose at C4. So our answer would be all of the above. Which of these are epimers of glucose? All of the above. Understood? I hope you understood epimerism by this detailed explanation. Right? I did not jump a single step. Anyway, you have any doubts? Pause the video, just go a bit back, see whatever I have said. Okay? Hopefully you'll understand each and every detail. So we have covered that epimerism as well. Okay. Now comes a very, very interesting thing, the pyran furan. Actually, to go to anemomerism, before we have to go to the pyran furan forms. Okay. So let's see. So whatever structure of glucose that we have drawn so far, those are linear structures. Okay. Linear structures. Open chain structures, which is never entertained by nature. Nature wants compaction so that things can be well folded, well placed in different cells. Okay. Our lungs have so many alveoli. Little lungs, but so much of alveoli saving so much of the surface area. Okay. Then the villi, intestinal villi. And the nephrons, and nephrons and kidneys. How are they fitting? Nature is a fantastic engineer about compaction. Similarly, these biomolecules also, they have to be compact to be fitted in cells. Okay. So this open chain structure that we see here, like This is what we say Fisher's projection. Okay. Fisher's projection. So he gave the idea about this linear, this open chain structure, which exists actually in nature in a very little form. Okay. So more compact would be a cyclic structure, basically. Okay, a cyclic structure. Cyclic structure looks like, ultimately it looks like this. This is the cyclical structure that we find. Okay, it's a heterocycle. Heterocycle because one cycle being formed of two different types of elements. We have the oxygen here as well as the carbon. Benzene is a homocycle because all the four elements are carbon there. Okay, so if this one is a heterocycle, oxygen being involved here. Now, how come this one falls into this? Okay, for that, I won't go much into details of the reaction mechanism because you don't need that for your biomolecules right now. But anyways, I'll give a hint how it happens. But how we achieve this, let's go step by step. Let us make this 90 degree. I mean, I want to explain this in such a way so that you can understand how it happens. Okay. Not even if you are not very confident about the reaction mechanism, but then also how the structure is achieved, more or less you will be able to figure out who binds with whom. For that, we also have to give the numbers. Numbers are very important here. So this is again our number 1, this is number 2, this is number 3, this is number 4, this is number 5, and this is number 6. Okay, let's first make it 90 degrees. Let's just bend it 90 degrees. Okay, fine. So that way, actually the structure becomes something like this. This one, again, I am telling you, this straight structure that it is given here, it is not that straight. There would be undulations, some, coming out, some going down, backward. Okay, it's a 3D structure, which we have generalized to make it a 2D structure, but this is a very random generalization. So ultimately, if we make it 90 degree, it actually comes to something like this. Let's see here. So first of all, this OH, this OH, this is our OH. So this is our carbon number 1. Okay, this is the carbon number 1. Okay, this is the carbon number 2. This is the carbon number 3. This is the carbon number 4. Okay. The number carbon number 5. I'm drawing only up to this. Okay, so let's see here. Fine. So this OH comes down. Okay, we have bent the structure like this towards 90 degrees. This H would come down and OH would go up. This OH would go up and this H would come down. This OH would come down. Okay, now comes the most interesting part here. What happens? It could have been that we were having the CH2OH here, CH2OH on this side. Okay, but if the CH2OH was on this side, actually what would have happened is that that CH2OH, that OH would have made a bond with this carbon. Then instead of a 6-membered ring here, we are seeing here 1-member, 2-member, 3-member, 4-member, 5-member, 6-member ring. Instead of a 6-member ring, we would have actually got a 7-membered ring. Had the CH2OH come here, okay, then that CH2OH, this 6 carbon, this had it come here, then this OH would have formed a bond with this carbon, basically covalent bond with this carbon. And then we would have got a 7-member ring, which is actually thermodynamically less stable. So basically, what was achieved was that this OH came here. Okay, this OH came here. The CH2OH remained here. And H was in here. Fifth carbon. So this H is here. This OH is here. Okay, this OH, this OH. I have just shown the bond that this is the purpose. I'll show you. Okay, this is how it stands now. Okay, this is how it stands now. Now what happens? I'm very short. I'm just explaining the reaction mechanism. What happens is that, so this will have its two non-bonding lone pair of electrons, and this would be our delta negative. Okay, delta negative oxygen, and this carbon will be delta positive. So what happens? This will donate a lone pair of electron to this carbon. So thereby, that forms a bond. Okay, that forms a bond. And subsequently, this oxygen also had its lone pair of electrons, fine. But this oxygen takes two more lone pairs and turns negative. Turns negative. Okay. As this one turns negative, and this one already donating its lone pair, now this oxygen has become positive. As it has become positive, now it will break this covalent bond and take the two lone pairs on itself. As a result, this bond is broken, and this hydrogen ion, now this hydrogen actually becomes an H+ ion, which will now get attracted to this one. Okay, to this one. Because this oxygen has turned negative. So this H+ ion will get attracted to this one, and now this will actually get the share of these two electrons to form a covalent bond between them. Okay, thereby the final structure that we achieve is, so this H is not anymore here. Okay, so you see the O is now O. O is now bound to whom? O is now bound to C1 and C5. O bound to C1 1 here, this is 1, and this is our final, right? The final structure is this one. If I draw a bit bigger, let me just draw that one. It will be easier for you. So, carbon. We have one H, OH, H, OH, OH, H, 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 C5 with one H, okay, and as CH2OH. Okay, so basically the link is between this carbon with this oxygen. Okay, so this oxygen will be attached to C1, fine. And then, I'm sorry, this oxygen will be attached to one C5, and this oxygen will bind to the C1. Okay, so the main game is about this oxygen of the 5th carbon. The number 1, this is C1, this is our C2, this is our C3, this is our C4, this is our C5, this is our C6. So C6 was always out of the game, right? Always outside the cycle. So it still remains outside the cycle. This structure is thermodynamically far more stable than being a 7-membered ring. Okay. So this structure, we call it the Howard's projection. This was Fisher's projection. This is Howard's projection. Understood? But something very magical has happened here. As we get a cyclical structure, something very magical has happened. How many asymmetric carbons, how many chiral carbons did we see in glucose? 1, that means at the C2, C3, C4, C5. But as soon as it became a cyclical form, we developed a new chiral carbon. See? Here, the C1. On one hand, it has H, right? On one hand, it has OH. On one hand, it is attached to this oxygen. One hand, it is attached to this entire thing, rest. So we have one new chiral carbon here as soon as the cyclical form happened for the glucose molecule. Right? So with this, what is the new game? The new game is that we have seen so far around chiral carbons, there can be rotation of the groups. So H can be here, H above, OH below. Or it can be that, so let me draw it like this. Either it can be H, OH, I'm drawing this repetitive, okay. Or it can be OH, H. Okay. So this is our alpha form. This is our beta form of what, sir? What? Alpha of what? Beta of what are they? So we are rotating only one. We are rotating only one. So it becomes epimer. So they are different compounds. No. This is again a miracle. They are not different compounds. We have made a cyclical form. That was an investment. So they did not lose their identity. This is also alpha glucose. This is also beta glucose. Okay. Alpha glucose, beta glucose. Both are glucose. Both are anomers of each other. So rotation around the new chiral carbon gives them same compounds, but different anomers. So this is another form of isomerism. Okay, but if I leave it just here, if I leave anomerism just here, then there would be a blatant lie. So basically, when you have to compare the anomerism, you have to compare the position of this OH with respect to this CH2OH. So here, what do you see? Here you see OH and CH2OH are on the same side, right? Then it's a beta form. And here, OH is downward, CH2OH is above. Then it's the alpha form. Okay, then it's the alpha form. Now let me tell you, if somewhere you get a structure like this, where OH is above and this is below, then do not just blindly seeing OH above say that this is a beta form. No. Here OH and CH2OH, they are on crisscross planes. Okay, they are on opposite sides. So this is also one alpha form. Understood? Okay. So we have covered each and everything about the furan we have not discussed. Okay, I'm coming to furan, but anomerism we have already seen. The furan form we have already seen. Okay, furan form we have already seen. Now comes the furan. Just like glucose here is a 6-membered ring, including the oxygen. There can be something like a 5-membered ring. Let's come to something like ribose. Okay, ribose. It's a 5-carbon compound. It's a pentose. Ribose is a pentose. And we all know ribose is present in the RNA. Ribose is a pentose. So again, how many chiral carbons? Here the chiral carbons would be 3. Say 2, say 3, say 4. Okay? So this is our what ribose? Very easy, D-ribose. Penultimate carbon, this one, OH is on the right. So this is the one, D-ribose. Okay? Again, this D-ribose will cyclize from the Fischer structure to the Howard structure. As it makes a cycle, the ultimate thing, the structure that comes out would be this. Wait. And there's no confusion because all OH are on the same side. HOH, H, and this is our. This always was outside the ring. So CH2OH remains outside the ring. Oh, how many members is there in? Including the oxygen, 1, 2, 3, 4, 5. So furan is this is our what? D-ribofuranones. Furan form of this pentose. D-ribofuranones. Have we still completed the full name? No. Where is the H and OH? OH and CH2OH are on different planes. So this is our alpha number. So this is our alpha D-ribofuranones. Just like here. Here we have this structure is our what? It's glucose, right? What glucose? It's a change from the D-glucose only. Cyclization of the D-glucose only. So obviously it's our D-glucose. It's a pyran form, 6-member form. So D-glucopyranones. But still we are not complete because OH and H, as CH2OH, they are on the different planes. So it is our alpha D-glucopyranones. That's it. So we have seen all the types of isomerisms in the carbohydrates, the aldose ketose, the diol stereoisomers, the epimers, the anomers, the pyran furan. And we have known who are same compounds, who are different compounds. Okay? So study this one very well. So in our next lecture on the carbohydrates, I will also give some sort of problems based on this. Okay? In that lecture on carbohydrates, we will deal with more of our polysaccharides. We'll deal with the different conjugated carbohydrates as well. Okay? So here we end today. Hope you liked it. Hope you learned something new. Thank you very much.
Dear students, if you ever feel that we are adding any value to your learning, then please subscribe to our channel and press the like button for the individual videos. This will encourage us to post more quality content in future.